coming along. And
thank you very much, Trevor. Thank you. And it's a pleasure to be back in Sweden. I suspect some of my genetics have roots out here somewhere, by the look at me anyway, but I always feel very comfortable in Scandinavia, and lots of very good friends here too. And hi, Gunnar, I saw you online. Good to see you. And we go back a long way too. Yeah, I mean, our first neutron deuterium scattering paper was 1982 with Andrew Miller. So we go back a long way. So the deuterium was very instrumental with not only neutrons, but NMR as well, which is where we had a huge number of advances and over the rest of the world. So it's great. So, I mean, I'm now retired. Oxford throws you out. I've had to sell all my magnets. I don't have any NMR facility. I've had to close down my labs and throw away all the PhD theses. So I've been writing up work that's been ongoing throughout the last decade or so, and trying to put all that together, and still writing something. So in fact, I submitted a paper 2 days ago. So I'm still a bit active with that. And then involved with the International Union of Pure and Applied Biophysics. So I'm still giving talks in various places when people invite me. And I'm delighted to be back in Sweden, as I say. I've been here before. There was a drug meeting here, so I came and gave a talk at it. So I started my career as a biophysicist in the biophysics department, the Ashby department. That went on from the Braggs. So diffraction was preeminent, and remember solving the crystal structure of sodium chloride and doing that by hand and by eye with no computation, of course, in 1968. But now, interestingly, having not touched crystallography all that time, I've come back to crystallography, and I'll tell you the story of that. So as part of this, we've been looking at water in receptor function, which is becoming popular with lots of different methodologies, not least structural methods where water becomes more visible. It's always been there with NMR, of course, in different ways, either as a hindrance or a help. So we've been working on this in the background for a long while, but now all of a sudden we can see water in receptors. So I can, is the range a bit too far? Yeah, okay, go back one, sorry. Okay, so, sorry, the pointer is, yeah. So here is something, it's only from literally about 5 years, about 10 years ago now, but this is a kind of a, from Martina Habenet in Bochum. It's one of her kind of cartoons showing how important water is along various timescales, and that's important along the bottom here. Because NMR has the capability of addressing individual timescales. Crystallography, of course, is only really applicable if things are motionless. But then solvation, kind of things we've been looking at for a very long time, decades, are important over longer timescales. So water is implicit everywhere, but can we actually see molecular water within some of these biological systems and investigate their importance? And so that's where I'm going to go to towards the end of the talk. Right, good. So photoreceptors are the most abundant proteins on Earth, and they are in every life form, in one form or another. And we've been working on these both from mammalian systems, but also archaeobacterial systems for a very long time. And here is a phylogenetic tree. And everybody will know about bacteriorhodopsin from Richard Henderson's structure and others. And the purple color you get in these salt beds as the salt beds, the salt pans concentrate up. And these archaeobacteria exist in this salt. You don't find archaeobacteria in rock salt, which is laid down before marine salt, because these evolved about 400 million years ago. And of course, rock salt was deposited way before that. But it's a very good model system for many biophysical systems as well, and many methodologies have been developed on this, first simply because of its structure and well-known function. But then, of course, we have retinal membranes. They evolved about 700 million years ago. So interesting debate about convergent or divergent evolution here. And the similarity in the photoreceptors in the retina at the back of the eye is, of course, the 7-transmembrane domain. And that is common across all of these rhodopsins. We've exploited the rhodopsins both in the retinal ones, but also in the archaeal ones. For example, we have taken individual bacteriorhodopsins, we put them on a gold surface about 2 by 2 microns and produced some very sensitive optical detectors. And they are more sensitive, more efficient than you find even in man-made devices. And now they are implanted, in fact, some of these things, not ours, but others, in the back of eyes to cure blindness in people who have retinal pigmentosis. And this company in Lund Division in Boston is doing that, in fact, with bacteriorhodopsin. So what's the common theme here about these photoreceptors? The common theme is retinal. Discovered by George Wald in Leeds in 1939, given the Nobel Prize for it. And it's just simply the isomerization of the chain of retinal within the protein that allows it to receive protons at a very high efficiency. A single photon is, in fact, activates this and then induces in GPCRs, for example, up to about 10 to the 6 impulses. So it's a very versatile but a ubiquitous molecule. And I'll come back to more about retinal in a moment. So what I'm going to do is talk about one member of these photoreceptor family, archaeorodopsins. There are 4 in this particular family, 1, 2, 3, 4. There's no crystallography for 3, and I'll come back to its significance in a moment. They pump protons similar to bacteriorhodopsin. You might think, well, why do we need any more structures of these types of proteins? It comes from a different genus. You find it in Tibetan salt lakes, and it's just evolved differently at a higher altitude compared to bacteriorhodopsin. But it has a very high sequence identity, and the 90% similarity is with the other AR proteins. So it's a family on its own. They do contain an additional chromophore, and that's bacteriorubrin. And the suggestion has been that it somehow facilitates the trimer formation of these proteins within the membrane. I don't think that's really true, simply because bacteriorhodopsin doesn't have it, but that forms a trimer. But it was not known whether these proteins are trimeric. So the organometric state of this protein was not known when we started this work, about a decade ago now. It's very easy to produce, as bacteriorhodopsin is in purple membranes. It's a red membrane. You can produce large amounts of it, fortunately. And there is a crystal structure, as I say, of AR2 at about 2.5 angstroms. And here we show the chromophore between the trimers. So why should we bother to even look or investigate another one of these AR3? Well, it's an interesting member of the family because the proton pumping, if you look to the left here, the photocurrent for bacteriorhodopsin, which is in purple, is about 1/10 of that for AR3 in a similar experimental setup. And this is from Boyden's lab. So it's a much more efficient protein, the much more efficient proton pump than bacteriorhodopsin. And what we want to look at is see whether that really is the case and why at a molecular level. It has a faster photocycle, although we don't have all of the intermediates. And also, it is used in about 1,000 labs worldwide as an indicator of neural impulses. And here this is a rat hippocampal neuron, and this is mapped at a fairly fast time scale. You can see the movie running. And this is optogenetics, and the optogenetics laboratories have been using this protein for quite some while now, even without the structure of the protein. And people like Ernst Bamberg, who examined the thesis this work's based on, and Boyden are leaders in this field. And I suspect that's going to be recognized more highly soon, I hope. So it's a protein for which we have no structure, but is very universally used technologically in optogenetics. So there is some interest in this. So no crystal structure. Some Fourier transform infrared experiments suggested different hydrogen bond networks compared to bacteriorhodopsin. People always compare it to BR, of course. There are conformationally distinguishable photocycle intermediates. They have been poorly characterized. There are short-lived ones, but they're also long-lived. There's kind of an incomplete photocycle there, showing some of those that have been resolved by a spectroscopy. So we know a little bit of information about it. When we characterize these membranes, we did it with the usual biophysical techniques. For example, dynamic light scattering of the claret membrane fragments. We don't think there's any significance here, but they're slightly larger than they are with the, slightly smaller than they are with the purple membrane of bacteriorhodopsin. The absorption spectrum it confirms the presence of the bacteriorhodopsin extra chromophore. So the purple line here has 3 maxima to the right, to the left, and that's for the whole membrane. And if you then extract the bacteriorhodopsin and delipidate that part of it, then the spectrum in black underneath is very resembles that of bacteriorhodopsin. So the bacteriorhodopsin chromophore induces this extra spectroscopic property on the protein and also makes it red, of course. As you might expect from circular dichroism at the right, you've got about the same alpha helical content as you have for bacteriorhodopsin. So there's a lot of similarities here. Now, if I may remember, I'm quite difficult in respect to my graduate students because I insist that we always work on functionally competent proteins. There's a lot of structure out there that is from functionally incompetent or really meaningless type of mechanistic details, often from crystallography. So I insist that the proteins we work on are functionally competent, and then you get the most information, but it can be very challenging. So one way of trying to determine proton translocation with this protein is to put it in bilayers and then irradiate with light and see if you can get proton translocation across. Now, Hagen-Bailey is in Oxford, so on talking to him, we thought we would try his droplet interface bilayer approach. And it's a new method for looking at transport across membranes from one compartment to another compartment. You may know this technology for DNA sequencing using hemolysin. It originated in the same kind of technology, and that, of course, was floated with Oxford by Nanocor at a huge amount of money not so long ago. So we took the protein across to Hagen's lab. You can see on the left here is a cartoon, on the right is the actual experimental view of that. And if you can see here, the trace in purple goes up when the light is turned on. So the light green shaded area is when the light is switched on. And then when the light is switched off, you get a drop in proton translocation. So it's a functional protein in these bilayers. If you compare that with AR3, you'll see that the rise in proton translocation is not only much faster, but to a higher level under identical conditions, and then drops off. And it's repetitive. You can do it here, it says twice, but you can do it repetitively. And in this setup, it's about twice as, 2 times more efficient than bacteriorhodopsin. So it's functionally competent and active in this setup. The gray line is simply with no protein because you need to be aware of piezoelectric effects and so on. So we do have a functional protein in this droplet bilayer interface structure. And this is just protein that's taken out of the membrane. Now, I didn't know whether any NMR people would be here, but I've thrown in 2 or 3 NMR slides for, to illustrate something. But membranes can often be looked at either as random orientations or as aligned membranes. And we've been developing a lot of methodologies for looking at aligned membranes in the past, not least because we want to exploit the anisotropy, the kind of thing that deuterium, for example, is very useful for. So you get orientational direction information from an oriented membrane. You can do this with bicels as well, and we worked on that a little bit, but it's mechanical orientation that I'm going to talk about. Now, this is with bacteriorhodopsin. This is with Niels Nielsen and Thomas Fosgaard at Aarhus University in Denmark. If we now label up, and again, the labeling was quite important, and here we've labeled all 9 methionines in bacteriorhodopsin. I'm showing this as an example to demonstrate something. And here, depending on what kind of NMR experiment you do, and I can talk about these if people are interested, you can either amplify the helices and the loops by the different pulse methods you use in the NMR experiment. I won't go into that in great detail at the moment, but this benefited from this labeling within the system. So, interestingly, you need to, of course, identify each of the individual methionines. We did that by mutation fairly straightforwardly. So we could pick out orientation information about all of those methionines within the protein. We could give some fluctuation information, conformational information throughout the protein at selected sites. And of course, you could do it with any others as well, but this one was particularly interesting. And we've done this for the loops of bacteriorhodopsin. Very often crystallography ignores loops, or loops are kind of drawn in because the resolution is not high enough, usually due to conformational disorder or dynamics. Now, of course, intrinsically disordered proteins are becoming important. So we did this for a membrane protein and found, in fact, that for bacteriorhodopsin, even the highest resolution structures in the database did not correlate with the membrane-bound, non-detergent solubilized crystalline proteins. So there's a lot in the loops, and I'm going to come back to the loops at the end for a reason. So if we take our NMR data, which is the top left-hand here, experimental pi-semer as it's called, and then we compare it with the highest resolution structures that were available. One of these is electron diffraction in the membrane, and the other are from lipid cubic phases, and this is a bicell crystal. And we compare those computationally. The best fit is between NMR and X-ray is in membrane. So the important thing here is that crystalline proteins, membrane proteins that are not in the membrane, sometimes have perturbations that you don't see because of the membrane environment is missing. So that was a lesson to be learned by that, and I'll come back to that again later. So the membrane embedded protein is that one. So the photocycle of bacteriorhodopsin is well known. And when we look at our NMR data, and again, it's very simplified here, there's a lot of work behind all of this. If we look at the protein in the ground state, or as a photo-induced intermediate, we can see that the lines shift, the NMR lines shift, but the greatest shift is from methionine 163. So that is undergoing a larger change within the protein. So we're looking at protein activation in a membrane directly by NMR. And it happens to be that the methionine 163 is in the loop here, in the EF loop. So that's telling us the EF loop is moving and changing the loop environment there. And again, I'm going to come back to this, so this is background to that. So the most sensitive residue is that which is helix EF loop in the 7TM. And it's also the most conformationally important loop within GPCRs as well. So there's clearly some importance there. And then just to finish this off, really the kind of classic cryo-EM and low resolution structures for rhodopsins, bacteriorhodopsins, you don't see the retinol. That has to be modeled in from a residual electron density, which is often much smaller, much lower than it is from backbone refinement, of course. It's the same for ligands. We did this for about a dozen drugs and ligands with drug companies, some of them based in AstraZeneca, for example, at I think Göteborg, who are funding some of this work in the past. So looking at ligands within membrane proteins, using NMR directly, using isotope labeling if necessary, can be very informative. And now you can conformationally define a ligand and then model that back into your low resolution protein structure. Now here, with bacteriorhodopsin, clearly there's no ligand visible within this cryo-EM or in the crystal structure. In fact, the first crystal structure of rhodopsin from that was produced was the retinol was modeled back into the protein using NMR data. So retinol is very important in these systems. I'm going to focus on that now. And it's clearly the isomerization here. Now by labeling up again with deuterium on the retinol, we can see some of the conformational changes within the retinol, within bacteriorhodopsin, within rhodopsin in this case, and this is bovine rhodopsin. We've done it for bacteriorhodopsin as well. That's all published and so it's a really important and interesting way of really probing into the active constituent of the receptor. In this case, it's the retinol. And again, this is all background for what's coming. I'm going to skip that one and skip that one. Just to show the resolution that you can obtain for these kind of ligands within membrane proteins. This is the retinol that's within, in this case, rhodopsin, bovine rhodopsin. And you can see the very high resolution distance measurements for the chromophore in the ground state rhodopsin in this case. And you can, this is really possible because we're looking at dipolar couplings between these labels, C13 labels within the retinol. So if you can label it, you can start to do a very high resolution distance measurements, and you can try to resolve what is happening to the chromophore with the protein during the photocycle. again, this is coming. So this was all published, and Gerhard Grosvenor is in Umeå now, who drove this work when he was in my lab in Oxford. And here we could see directly the isomerization of the retinol from 11 cis to all trans within the protein on isomerization. All of that was from direct, independent, not model dependent. It's all direct measurement of ligands, and in this case, retinol within the protein. So back to AR3, a bit of background there to other things that we had done. But now let's come back to AR3. So I'd already mentioned to you, EF loop is very important in the activation of these proteins. And if this is some high-speed AFM we did with Ando and Miki Shibata in Kanazawa, and here you can see quite clearly that firstly the protein is trimeric from these images on the right, and then when the light comes on, you can see the protein moves within the lattice. The light comes on, the green bar comes on, and the protein is actually moving within the lattice during the photoreception. And they have done this previously with BR, and now this is the only other example of which they've managed to do this. And here on the AR3 membranes, you start to see a center of mass change. And of course, this, and I showed you earlier from the NMR, is because this EF loop is undergoing conformational changes and relaxation of the loop, in fact, around the methionine, which you also find in the AR3. So the NMR told us this some years ago. Here we have it directly by AFM and visualizing it within the membrane. So these are real-time changes as well. Now, I said we could get hold of tons of membrane. That's literally true. We use the conventional lipid cubic phase of the protein, the membrane fragments here, top left, you can see it in Eppendorf, and then crystallizing the protein. This is the old Lazati model that I'm sure Trevor remembers from decades ago. This is now very important. Martin Caffrey, for example, has pushed this to membrane proteins, and many others now. There are many versions of that. And then the crystallization here we were doing with the Wellcome Trust membrane protein lab at Diamond with Isabel Morales. And we have a wide range of crystals that we've been able to form here in different shapes and sizes and forms. In fact, some of them are really rather large. And you know, if labeled, they could be useful for neutrons, for example, if somebody wanted to pursue that. But we can produce large amounts, milligrams and milligrams of nice fine crystals. And that's going to be important when we come to the last part of the talk. Now, clearly, diffraction has been pretty good. We can use molecular replacement, of course. And so what we have now are ground state structure at 100K of about 1.07 angstroms. And then we've got a dark adapted structure. There are not many dark adapted structures in the database for photoreceptors. It's a bit tough to do. You have to put it in a dark, you leave it there for an hour or so, and then you have to do all the crystallography in the dark. So it's a bit less easy to do. But we managed to do that and get a structure at 1.3 angstroms. So it's a fairly respectable resolution for a membrane protein, of course. And all that's published, and down here is one of the references for that particular work. We've got a lot of structures now. Some of them are creeping into the database bit by bit, but they take a while to get in there. But there's probably about 5 in the protein database at the moment, and there'll probably be another 10 as time goes on. So we have some high resolution structures. We didn't use detergent. There's no detergent with this protein at all. I've already said talked about detergents and how much they can be perturbing. So all of this was done with membrane fragments directly from the cultures, and then adding the lipid cubic phase to get the crystals. But we also, since we have all the endogenous lipids around, and there are about 10 lipids for each protein in these membranes, there aren't many. So we were wanting to analyze that lipid component because it had not been well done. And so we added back some lipid, just a saturated lipid to that. And then we formed these lipid nanoparticles here. And this is the styrene acid, which we've been working on again in parallel, again for about 15 years or so now. And it's not dissimilar to lipid nanoparticles used so clearly for COVID vaccine delivery, but Peter Cullis's mixture is slightly different. And then the protein is embedded within these kind of 100 nanometer diameter particles with the endogenous lipids and some of our saturated lipids that we put in. And then with Carol Robinson at Oxford, we took our samples across to her and said, would you do some mass spec on these samples, please? And sure enough, we've done electrospray mass spec, and they seem to work. They seem to work pretty well. And in fact, if you look at the data here on the right, you can see the purple squares. There is AR3, and AR3 with one of our endogenous lipids we added back. But you can also see AR3 with a green dot, which is with one of the endogenous difunctional lipids. And then one of the sulfonated lipids in blue is there as well. So we could identify the individual lipids here, and then a repeated mass spec can release those lipids from the protein. Now, what's interesting technologically here is that this is at very low voltage. This is the kind of conditions under which Carol would do regular soluble proteins under. Whereas with membrane proteins previously, without the lipid nanodisk, in this case, you'd get a broad hump, some spikes on, and by deconvolution and massaging the data, you could pull out narrow spikes. So you could characterize the individual components. In this case, that has worked perfectly well using the nanoparticles. And now her new company, OMAS, I think it's just been called OMAS Therapeutics, they're using this technology for a wide range of drug targets, including GPCRs. So these lipid nanoparticles are really revolutionizing mass spec of membrane proteins. So I think this is one of the first example of how that was done. So there's a big controversy in the literature about post-translational modification for bacteriorhodopsin. Is it post-translationally modified or not? Is the N-terminal modified or not? Now you might think, well, we could just do some mass spec, but that's not been not resolved. We could look at a crystal structure. Again, that's not been possible in the past because the resolution of the N-terminal has not been there. So we thought we would look at this for this protein, given the resolution that we have. And you might imagine the signal peptide here removed, revealing the glutamine, and then of course you've got modification at the end. Does that actually happen? So you might hope that the glutamine here is then converted to the pyroglutamate, and then that should be resolved. So if we look at the crystal structure, these two potential N-terminals of AR3, we should be able to see whether that has happened or not. And sure enough, here is the electron density around the N-terminal that we can resolve so well in this structure. And there's one of the particular database numbers, and you can see it clearly is the pyro derivative. So it's pretty obvious it is post-translation modified AR3, and you could probably do the same with bacteriorhodopsin if you get the high enough resolution. You might expect it to be post-translation modified, of course, simply because you don't want the degradation by the amino peptidase, but also lysine is incorporated into the protein as a shift base, and you don't want lysine forming a shift base with the terminal amino group of the protein. So that makes sense, and that seems to, for this protein at least, solve that particular question. Now, what about the retinol? Coming back to the retinols, which we studied by NMR so long ago, what can we say about the retinol within this protein? Well, until now, fairly poor resolution retinol data was used to model back the retinols, as I said, often done with NMR data to help it. But now we've got pretty good resolution of the retinols from the electron density directly. And you can see it's an all trans isomer here, two distinct conformations to the beta ion ring. Well, I showed you that earlier from NMR, and that we published 20 years ago now. And of course, the community was not really very interested in believing NMR data. Here we have crystallography data, so I think we've just confirmed what we saw a while ago. And the pocket, of course, the binding pocket for retinol, we can define that very well now. It's more hydrophobic than for BR. It's got a different color, as you saw, and that you can see quite easily here if you do the gravy analysis, because there are 5 more residues in the binding pocket. So we know a lot about the binding pocket now. Okay, so that's a light adapted form at the 1.07 angstrom. What's it like for the dark adapted form? Now, this was a bit of a surprise for the crystallographers, but NMR again, we showed that indeed the retinal is in two conformers within the dark adapted protein. It's much easier to do dark adapted protein by NMR than it is by crystallography. And these two conformers were fitted. You can see it here, at the top right, around the isomerization site, and the shift base is there. So indeed, this does have a mixed conformation within a dark adapted protein, and that stays about the same regardless of how long you keep it dark, and so on. So how is that the case? Well, we went to Igor Shapiro, who was in Israel. He's now back in Germany, to look at our structural data and see whether he could understand that energetically. What you see here from his QMMM calculations is that there's a lower activation for the cis-trans isomerism, so that's going from cis on the left to trans on the right, in the dark adapted state, which is the blue line at the bottom here. So the difference between the cis and trans is really quite low, even at cryo temperatures. And so the chances are that there's going to be an equilibrium between those two, given the delta, the thermal contribution to the isomerization. But the all trans isomer is more stable in the light adapted form. So in red here, between the cis and the trans, there's a much higher difference in the activation energy of those two forms across, and this is across, of course, the dihedral angle of the C13s D14. So that's telling us the C13 isomer is more stable in the dark adapted state than in the all trans, and that's what came out of these calculations. And so retinal does have a mix. It's ready to flip. It's ready to isomerize. And there is just a small difference between the two forms in the dark state. So it's ready to jump and ready to isomerize and then be activated by a photon of light and then transport a proton. So that was quite an interesting excursion into some energetics calculations. But then we thought, well, we know quite well the amino acid composition and position of all of the residues around the binding site. And there's been this theoretical paper in about 2012 suggesting that if you can shift the electrostatic environment around the chromophore retinal, then you should be able to shift its color. So if it goes red, it's a bathochromic shift, and that's by increasing the hydrophobicity around the shift base, and then a hypsochromic shift if you make it blue by reducing the electrostatic contribution around the binding site. So if we mutate some of the residues around here, we ought to be able to change the color of the protein. Bacteriorhodopsin is purple, and red membrane is red, but it's got the bacteriorhodopsin in it, of course. So can we do that? Well, we can, and the student involved with this now at MIT, he had made 23 of these different isomers. And you can see top left here, the absorption maxima of each of these different proteins. And bottom left here, you can see the different colored membranes. You can see yellow one, a purple one, just like BR, a green one that's almost colorless, and then another one which is going over towards red. And so we even produced two red shifted and blue shifted, a number of blue shifted mutations. So this is quite nice. We can tune the color. Bear in mind, man-made detection devices are either on or off. They're only really detected to wavelength and by filters and reconstruction of the triggering. And we did some of that work with DTSL, with the military people in the UK, because that was quite interesting for them. But in biology, we're sensing light. We're sensing different chromophoric absorption maxima in biology, which is really quite important to know. So having done that, clearly I said, well, is this functional? I don't mind seeing different colors, but does the thing pump protons? And sure enough, it does. Here are the two mutants, and the wild type here, then the blue mutant. This is the triplet of residues we needed to mutate to get that color change. And then here's the activity data. And here are the two membranes on the left, a blue one, a red one. And here you can see current output from the membrane using the droplet interface technology. So we do have a functional mutant of this protein, which is at the blue end. Now, why would we bother with doing that? It's not just an academic exercise. There is some interest here. So these reporters have a much lower background in brain tissue and could be imaged deeper. And the optogenetics people would really like these kind of indicators. So if you can change it, then you can be able to image deeper using their methodologies. But also, you could get a sensitive readout, and you could start to avoid crosstalk between other receptors if they're widely different colors. So now you could really kind of start to do some multiplex voltage imaging in these systems, trying to look at various neural signals and see how they combine and how they disperse. So multiple receptors of different colors based on AR3 could facilitate that. And I think people are thinking along those lines now, given this information and this possibility. Of course, we're not doing any more of this now. Right, towards the last part, why is it more efficient at proton pumping? So everybody knows the residues for bacteriorhodopsin. So I've enumerated the ones here based on bacteriorhodopsin, but just add 10 onto those for AR3 because it's got 10 more residues, 5 of them in the retinal binding site. But for ease, I've just put in the bacteriorhodopsin ones so you can see them and maybe relate back to BR. So the proton release complex is around here. It's been known for bacteriorhodopsin. I'm going to come to this arginine 82 in a moment. And we can now visualize into the protein at these important sites directly and try to understand why AR3 is that much more efficient than other photoreceptors. So what we found is this arginine 82 from this very high resolution data now is really in 3 conformers. And it has equal resonancy in the 3 particular conformers. And that has really come from the smeared electron density rather than the very well-defined in this particular part of the proton release complex. So if you now try to imagine and see that the water component in those environments, you can see that there are now 3 distinct pathways for proton translocation across the complex. So there are 3 different ways and multiple ways. So that was quite interesting to see. And I'll compare that with bacteriorhodopsin in a moment. And that really are defined by the orientation of the arginine. You can see it on the left, the middle, and then on the right-hand side here. And you can see how different they are. And of course, it's centralization, which is going to be determined by this hydrogen bond network through here and proton translocation. So the water network is really quite important here. If we compare that now with bacteriorhodopsin on the right, arginine 82 is highly constrained, and there's just one pathway for proton translocation. So that's a big difference. So there's 3 with AR3 and just 1. So imagine a single highway with a constriction and just single highway of traffic going through, compared to a 3-lane highway, and protons being able to go across the full highway through the protein. So that almost certainly contributes to the higher efficiency of AR3. Now, I mentioned very early on that there was this FTIR evidence that, and they said in their manuscript that there's a somewhat different environment resulting in a weaker hydrogen bonding in the light adaptive state of AR3. And that probably confirms it, but here we've done it specifically and identified precisely which residue was involved in that. And you can see about the interaction of the W406 as well. So if we now ask the question, why is there much faster photocycle? Well, it's pretty obvious when I mentioned the 3-lane highway that the pathways are much more open. But it's not the only difference in feature. if coming back to loops, which we addressed by NMR so long ago and showed how important they are from NMR, we shouldn't ignore them. And if you look at bacteriorhodopsin, which is in purple here on the left, you can trace the EF loop of bacteriorhodopsin, and I mentioned that earlier. Then if you trace them for the archaeorhodopsins, the AR family, they have a so-called omega loop. You can see an omega forming there for the AR2, now the AR3, which we have a very high resolution, and AR1. So why do they have an omega loop and bacteriorhodopsin does not? So why do they have the loop? Well, if you now look at the waters, this is the 1.3 angstrom resolution structure. You now see, I've enlarged this part of the protein around the loops. You can see there's a huge water reservoir here in the loop region, the omega loop region, this protein. So there's a huge reservoir of water in this loop region, which you don't have in bacteriorhodopsin. And now we have a wider pathway for proton translocation across the protein. So this almost certainly, these two things together contribute to the higher efficiency of AR3 compared to other photoreceptors and bacteriorhodopsin in particular, which we can do a direct comparison. So that, we think, explains that. And so we thought, well, you know, if it doesn't move, it doesn't do anything. So proteins move. You need to start to use methodologies, including NMR, which look at dynamics, but also now crystallography, of course, has gone that way. So you probably all know about serial femtosecond crystallography and XFELS and the developments in those methodologies worldwide. And of course, we've got the contributions from Henry Chapman and Richard Nutzer. And we have used this methodology with our AR3. The nice thing here, this is Soiwata's setup in Sackler at Spring 8. Here you can irradiate, in this case, a photoreceptor with a laser pulse of light, activate the protein from the dark state to an activated state, and then follow that by an X-ray pulse. And then, because of the nature of the experiment, get the structure of the protein as it's activated in different timescales. And of course, it's non-destructive in terms of or at least the structure you get out is before the protein falls apart. So it's a very authentic way of looking at structural changes. Now, for this, you need nanocrystals, and you need masses of nanocrystals. A whole hypodermic can last about 20 minutes on the XFEL setup that SO's got. And we went out to Sackler, and we had 3 36-hour sessions, fortunately. We were lucky to get that. And we carried out a large number of experiments on these systems. And so if anybody is nervous to strobe lighting, they should not look at this. I'm going to try and set this movie off, I hope, if I can. There we go. So now you can see the crystals top right there forming through the tube. The bottom right, there's a funnel which collects the crystals and they're sucked away. And then in the middle here, that's where the beam goes to carry out the diffraction. And there are 3 different views there, and you can just sit there and watch it. It takes 20 minutes to do one of these courses of experiments. And it was really pretty exciting to watch, I have to say. I was staggered by it. And I can go on one. Yeah. I think I've taken that one out. Yeah, okay, good. So just as someone who has known crystallography since 1968, 69, it was spectacular to have seen, you know, I did my processing on a FASIC calculator on paper and longhand. Here, I could sit in front of this data output, and they had done the data analysis directly, and I could see this kind of output on the right here in real time as it was happening during the 20 minutes. And the structures here are coming out at about 1.5 angstroms. So if you look here, this is a 5 femtosecond map. We've gone from 5 right the way through to 1.5 milliseconds. There's no light contamination in these systems, of course, it's totally dark. And what we can see here is that even after 5 femtoseconds, the water electron density is changing around the retinal and the chip base. So At this very early stage in this protein, water is absolutely implicit in the photon reception, one photon incidence on the retinol. So we can see that absolutely directly. And then we've got about 15 structures now, and we hope we can put them together. Others have done it with bacteriorhodopsin. This is with AR3. Interestingly, the protein is functional in the crystal form here, in the lipidic cubic phase crystal structure. It's no detergent in this, but we're showing that we do have a functional protein within the system. And it's D85 in this case where the initial structural changes are. I can't tell you any more about this. The data is still being analyzed, but that's where this has kind of ended up. So in conclusion, we've got purified crystal structure of AR3, a protein that hadn't been crystallized before. AR4 crystallizes too, but we haven't spent so much time on that. There are subtle differences in the retinal isomerization state, which now you can see directly by crystallography, had been seen by NMR 2 decades earlier. There are differences in the hydrogen bond networks of functionally important waters, post-translation modifications, and then the development of native mass spec membrane proteins has contributed from this. And then obviously the water residue mobility. It's a vital role in sensitization of the receptor. And I think, just coming to the last point, I suspect, and people like Gunnar may know more than me, because I've not been keeping up so much, but the generic implications for water in general, not in just reception, but in biological systems at this level, I think is going to be very important. And I hope that's given some indications. Just to thank the people. Peter Judge is a long-term postdoc in my lab, interacting with Trevor and his groups significantly. Javier's at MIT now. Juan is now a group leader at EPFL. Carol, of course, and Hagen and Diamond for helping us get there. And we did AFM with Pierre Emmanuel, and then also the energetic calculations with Igor and Mickey Shibata for more cryo-EM, the fast AFM. And this is Isabel Morales, who has really driven the crystallography. She's gone off to Google DeepMind, so I'm not sure how the data will go. So thank you very much, everybody.
Thank you very much.